I'm Robert Nathis. I'm a graduate student from MIT, and I work in the Gupta lab at the Whitehead Institute. My research at this point is primarily in cancer biology. And what I'm interested in is the differences between cells within one tumor. Tumors arise from genetic mutations. And it's actually a series of genetic mutations that result in a tumor. And while the tumor is developing, more mutations keep happening. And this means by the time a patient gets diagnosed with a cancer, there's a lot of genetic heterogeneity within their tumor. And one possibility is that different families that are genetically different, families of cells that is, could behave differently. And it might be this heterogeneity that's causing some of them to live after chemotherapy while the rest of them die. To investigate different families of cells within a tumor, I needed a way to tell that they were actually related to each other, sort of tracking their family trees into the future. So this was basically one method of doing that analysis. What I basically did was mark these cells so that they were given a certain color, such that this color was stably maintained through cell divisions, so that if I made a cell blue, all of its descendants would be blue, and if I made it purple, its descendants would be purple, etc. And that means that even in just a plate in the lab, I can look at a field of cells and then figure out how they're related to each other. And this is indeed what this image is. And it's pretty striking that even in this relatively homogeneous population, these are normal cells and they should be mostly the same, you can tell that after this overlay of familial relatedness is put over these cells, that cells that are related to each other look more similar to each other. Which, I mean, maybe doesn't come across as so surprising, but I was pretty surprised when I saw that. So the actual way of making the cells this particular color is pretty cool. So in the same way that you can make pretty much any color in a computer screen just by combining different amounts of red, blue, and green light, that's the exact way that I made these different colors. I just randomly made them express blue, green, and red fluorescent proteins, and these random levels of these proteins ended up making different colors.